approaches to study how plants respond to herbivores both above and below ground, with important implications from ecological theory and sustainable agriculture. Her talk is titled "What Goes Up Must Come Down: Interactions Between Above and Below Ground Herbivores via Induced Plant Defenses." Nicole, thank you so much.
So while my slides are being pulled up, thank you very much for the invitation and a kind introduction. I'm very happy to be here. I really entomologists are a nice community, so I enjoyed these days here with you tremendously. Not in the least also the dancing yesterday, so thank you for organizing that. Right, seems to be working. I just need this pointer online. Can you pull this up for me? You know that you can use the mouse to point so people online also see where I am. If I say point at something, they don't have to guess. Oh yeah, that's maybe here somewhere. Yeah, otherwise I will just say, no, that's also not it. Maybe just start it and we'll be fine. You just start it. And I think it's down here in the corner here. There. Laser pointer. Yeah, thank you very much. So thank you very much again for inviting me. Also welcome to the people online that I hope, oops, it's going, yeah, okay, it's going away. I just want to also start with, as a director of an institute, of course, I'm doing more administration and scientific, hands-on scientific work. So this is very much the work of three of very talented PhDs and now partly postdocs that are working with me, Jessel, Rebecca, and Axel, that you see here on the picture. So, see this one. So page down also doesn't work. Okay, it does work. Maybe I should press a bit. Okay, a bit more. Yes, okay, thank you. So assuming that not all of you are very much aware of how plants respond to herbivores, I will give a little introduction on induced plant responses, so we all on the same square before I start to talk about also how can these plant-induced responses be causing interactions between herbivores that are feeding on the same host plant but never see or meet each other. And then finally, I will give some examples from our own kitchen, basically the work of the people I showed on the picture before, where I show also not only on above below ground, but also between root feeding herbivores. And our kitchen and also in my past, but also in Groot-Beren, is very much dominated by Brassicaceae. They are very diverse in their shape and flavors. As you see here, we eat them maybe almost every day. And this is what I will talk about. The research is all on Brassicaceae plants. So induced resistance or induced plant defenses were actually more or less discovered by scientists when I was about to start my PhD in Leiden, so around the 1990s of the last century. Sounds very far away, right? And people were also very skeptical about them. But what it comprises is that plants are, when they are attacked by herbivore, as you see here, this little caterpillar biting in the leaf, that they start to actually, there is a cascade of responses that usually also involves plant hormones. I will come back to that a little bit later. For example, jasmonic acid, and these serve as signals for the plant to start defending itself. So in that case, or in the best case for the plant, maybe the worst case for the caterpillar in this picture, is that there will be toxins made, and these toxins immediately kill or deter the herbivore. And this way the plant can reduce the damage. And what is important here, that you have local responses. This is where the herbivore was feeding. And there is also where the toxin and immediately affects the herbivore. But you also have systemic responses. So the responses don't stay only in the place where the herbivore was feeding. So there's also effects in undamaged organs. So induced plant responses can be local and systemic. And what is also very interesting, and what is a lot of when people started to think about why are induced responses so advantageous, because you always have already some damage, is that they can be very specific. So you don't have to defend against any potential herbivore that may come by, you just only defend against the herbivore that you perceive. And what is also an important thing is that that these hormones, so there's not one hormone involved, there's multiple hormones involved, they can crosstalk, and this can also affect the response of one herbivore to that is feeding on the same plant as another herbivore. And so this was also then a realization. So if these responses don't stay where they are, they may also affect other herbivores on the same plant. And if you're maybe aware more in population ecology or in insect community ecology, sometimes people talk about apparent competition, and sometimes apparent competition is maybe caused by induced defense responses. Yeah, so how do plants now know? That was also first like very skeptical. How can a plant know there is caterpillar A or there is something else feeding aphids? People were really skeptical about it in the beginning because you consider plant as very passive. Well, we know now that there is elicitors that can increase the damage. I see a little box here. Should I move that? It's not on the way. Yeah, that there is elicitors and that can trigger this cascades, the signaling cascades. So on the one hand, is damage. So there is some cells are being damaged, and this releases, for example, cell wall fragments, and that you see here, for example, on the right hand side, oligosaccharides that could signal to the plant your damage, do something. Like if we have a wound, you have to close it, otherwise you bleed to death. Same response basically as plants, but also herbivores bring with them specific elicitors, and this is something that could be in their example in the regurgitate or saliva, and this is something that herbivores then that the plant is exposed to while the herbivore is interacting with the plant, and also gives the information about who is feeding there. So it's very well known that salivary compounds can do that, but also pheromones, or if you have ovipositing insect, oviduct secretions can give away about, is this a beetle or is this something else? Is this a sawfly putting an egg on the plant? Yeah, there's also something else which is also very relevant to my talk, and these are effectors. And elicitors, as I said, are things that come with, are cues that come with insects, but are not necessarily have a function of giving them away to the plant or for the interacting with the plant. Sometimes they're, saliva is needed to digest, or obviously, but it's not something that the herbivore can suppress or do something about. They sometimes just need it. But effectors are cues that are specifically used by herbivores to interact with their host plants, and often it is to suppress the responses. And the two organisms that I will talk to about today, Mysis persicae, aphid, and root knot nematodes, are examples of organisms that have a lot of these effectors and are manipulators of the plant defense response to their own advantage. So, for example, for aphids, you know that they have stylets. They feed by moving the stylets between the cells. Sometimes they do little probing. So you see here that they test some cells on the way to the phloem where they start feeding. And what they inject with this, so apart from the sheet that is also guiding their proboscis into between the plant cells, they also inject effectors. And they do this to tinker with the cellular signaling. So they want to suppress the response of the plant. So the plants would, in an effective defense response, the plant would maybe block off with callose this phloem sieve element. So there is effectively no nutrients coming to this aphid again. But the aphid, of course, would like to prevent this because otherwise it doesn't have a feeding site. So these effectors are specifically injected to manipulate this and create a feeding site, a sink for the aphid to stay on the plant and stay basically, be able to feed there. So the other category of herbivores, some people call them pathogens, you can just debate about it, are nematodes. And these are often overseen, very small worm-like animals. I've heard several talks already. People talk about entomopathogenic nematodes, so those that attack insects that will also use in biocontrol and that very effectively sometimes. But today I will mainly talk about the other categories, so root knot nematodes or cyst nematodes and root lesion nematodes, which are plant pathogenic nematodes. So they're like the entomopathogenic nematodes. They're very small worms, basically. They are very omnipresent in soil. So there is high numbers of these in the soils. So the basic teaching book from Wageningen University say there is no plant without a plant pathogenic nematode. Of course, it's hard to prove that, but they're very omnipresent. They're also highly diverse. So there are many genera per square meter found in fields. And they can also cause severe crop losses. I think that one of the talks before here is like in the potato, it's like 27% of the pesticides in potato were used against cyst nematodes, which is a big problem. So today I will talk about two of these. One is a mobile entomopathogenic nematode, Pratylenchus penetrans, which is staying in its worm-like shape. It's also penetrating in the roots and they're feeding on cell contents. But it's not like the other nematode, the root knot nematode I will also talk about, is not sessile. So it will stay mobile and basically makes a lot of damage, which causes lesions visible on the roots. So the sessile entomopathogenic nematodes like Melodogene incognita, they form, so the females form a root knot, they establish there, they make, like the aphids, they also create a sink there and they will stay there, produce eggs and then die. Yeah? And the eggs will be released in the soil. Juveniles will emerge and the cycle closes. And as they're aphids, they're above ground counterparts. I always see nematodes and aphids, they're a little bit, they're above and below ground counterparts of each other. They also inject effectors in the plant. So what you see here on this picture, in this diagram, is that this is the head of a nematode. They have a stylet that they insert also eventually in the cells. They create specific, they also manipulate the division of the cells, so they create a really big knots or super cells, as they sometimes call, to feed from. And what you see here, all these little balls and squares are also these effectors that are meant for first of all, to suppress the defense response, otherwise the plant will recognize as a pathogen and kick it out again. And also to make these root knots. So they're also masters of manipulation. I think there's hundreds of these effectors found in genomes of nematodes. It's very hard, of course, to collect them because they're so small and there's so little of them. But with the modern molecular technique, people get a lot of insights on how many they have. By the way, just for the record here, they have also called amphid secretion, and that is meant also for them to survive and take up nutrients. That is again an elicitor, just to show that both are there, but they have different origins, so to say. One is there because the organism cannot survive without it, and the other one is made by the organism to interact with the plant. Okay, so I also talked a little bit about plant hormones. So this is your basic plant physiology, taking you back to maybe botany or plant physiology 101. So we have different plant hormones that you see here that are involved in growth and development of plants. For example, ethylene or indole acetic acid, auxins are very famous for affecting growth and senescence of plants. And in the defense responses, we mainly have been working with jasmonic acid, so which is induced, for example, by feeding caterpillars, but also by the cabbage root fly, I'll talk about later, and but also by some nematodes, where salicylic acid is known to be generally induced by aphid feeding, but also by these mobile Pratylenchus penetrans nematodes. So these are hormones that are specifically studied in the context of these induced responses. And now what is interesting also particularly is that I sometimes compare this with like a piano. Yeah, you have many keys, black ones, white ones, and all kinds of, and you can play different tunes on it. And this is also what hormones do. Yeah, depending on which ones are expressed, you have a different response. So it's not one hormone does one thing, it's the whole complex of all these hormones that decides what is the actual response that the plant will show. And it's very well studied by many people that when, for example, salicylic acid induced responses, for example, by a phloem-feeding insect like an aphid is being induced, this can also, and at the same time, there's a chewing insect and inducing the jasmonic acid response that via all kinds of interactions between also ethylene and abscisic acid induced responses, the outcome of a single infection or a single herbivore event can be different from those that are when there's multiple herbivores in the plant. And those of you that work in fields, also either with wild herbivores or with wild, or even with agricultural fields, you know there is never one herbivore or one problem or one pathogen. It's always the whole complex, yeah. So plants have evolved all these complex defenses to deal with this over the millions of years that they have been on Earth when we were still not, I think we still had to be invented, then they were still already dealing with very large herbivores like the dinosaurs, yeah. So they're masters of survival, and these systems that we trying to understand today are a result of million years of evolution. I'm always in awe of plants, how they did this. So, as I told you now, there is induced systemic responses. So one herbivore feeding on one part can affect another one. This has been very well studied and originally, of course, discovered by above ground interactions via induced responses, but can also, of course, happen between below ground and above ground, and also in below ground systems. And can have different outcomes because of these crosstalks between the hormones. Depending on who is feeding, it can affect the other partner, which is not interacting with this other herbivore directly via induced defense responses. And I'm trying to understand how this can work. And sometimes we see it's facilitation, and sometimes we see it's inhibition. And yeah, this is something we have been working on for quite a while, and it stays very interesting. So now let's go to, I hope you have now the basis to understand the rest of the talk, which is our own results. My title was about above and below ground, but I cheated a bit because I also did something on systemic responses below ground, so between a root chewing herbivore and a nematode. And then we also go to the below ground, above ground effects, all in brassicaceae. So let's start with the main course one. That's basically, this is the work of Axel Thau, who defended last year. And he was interested to understand how systemic induced responses can work below ground. And you say, like, what are you talking about systemic induced responses? This is all roots. Well, because roots are also below ground, many people don't realize it's not a root. You have main roots, you have side roots, you have fine roots. And organisms that interact with roots are not only very diverse, from microbes to large herbivores, but they also in separate compartments. Nematodes can only enter fine roots because otherwise the root is too tough. So they really enter at the tip and develop there and stay in these fine root area, so to say. Insect herbivores, like the one we work with, the cabbage fruit fly, they mainly feed on the taproot. So there is a kind of division of where herbivores and other interactions take place. So nematodes and insect herbivores may be in the same compartment if you look at it as the soil, but they are definitely not in the same subsection of the root system. So this is what he tried to understand together with, by the way, with Jessa and Rebecca, which are also in the picture. And the study system that he used is Delia radicum, which is a cabbage fruit fly, which is native in Europe, but it's also meanwhile in the canola fields in Canada and Northern America. It's invasive pest. Yeah, it has as a fly it has the maggots that feed on the roots, and they're mainly in the main roots, as you can see here. And we also have the root knot nematode in this case, which is one of the nematodes feeding on the fine roots of Brassicaceae species, and also can cause problems in many cropping systems. The cabbage root fly is a specialist, so it's feeding on many Brassicaceae. on the main root, and the nematodes are polyphagous, so they're attacking a range of crops, but including also Brassicaceae. And as I already said, they are in the finer lateral roots, whereas the cabbage root fly is feeding from the main roots. And the reason we really did also test, they don't like the fine roots. Probably it's not enough food for them. They really always go for the main root. So what our research question was, so we already knew some things. So we know that these plant parasitic nematodes can induce systemic responses also above ground. There was some older work that, but and they're very ubiquitous. We know that they're also in Brassicaceae plant. So we postulated that if you have already nematodes and then you add a root fly, there should be different responses, because if there are systemic responses in the root as they are above below ground. So these are the research questions. So if we infect plants with root not nematodes, do we see a change in the response or in the performance of Delia radicum? And what are the underlying molecular mechanisms? Yeah, so let's start with the performance. So the experimental layout is quite straightforward. We always started with the nematodes. Why? I think plants starts their life also with interactions with nematodes. As soon as the seed is germinating, the root is out, they go in the soil, there will be nematodes. Yeah, the root flies, they don't come till later, till the plant is a little bit bigger and has enough biomass for the mother to decide this is a good plant to lay my eggs. So we infested the plants with Melodogena incognita, 400 juveniles. Is a moderate infestation. Some people use 2,000. So those are not really the field levels that we find in wild plants. Then Axel put eggs of the Delia radicum, and he looked at the performance of the flies, letting them emerge, and collected adults and assessed their body mass. This is also, by the way, published this year in Plant Physiology, so you can read all the details there. So what we found is that if Delia radicum is being reared on plants with these Melodogena incognita, that they perform better. Yeah, so were we surprised? Yeah, a little bit. I thought it was quite a strong effect considering. And they didn't only have more adults emerging, which you see here, that more of the eggs came back as adults in the end. But we also saw that the adult weight, so the darker distributions there are the ones of the plant on with already nematodes. And of course, you see there is a sexual dimorphism where females are heavier than males, but you see in both cases, the ones that were developed on plants with Melodogena incognita were heavier. That's of course a very strong potential fitness effect that there will be more eggs in the next generation. By the way, Delia radicum has 4 to 5 generations per year, so it really also adds up. If you have this in your crop, your population will be much bigger. So we thought this was, of course, very interesting, that there would be a positive effect. We also, of course, looked at the mechanism. So Axel did a series of experiments, which I, due to the time, cannot explain here all today. Like I said, I happily refer to the paper. We looked at phytohormones, primary metabolites, because also primary metabolites may have an effect on insect performance, and defense metabolites. So let me jump into these mechanisms first. I will skip the phytohormones because the patterns were not so very clear and easy to interpret, although we saw some nice effects. First of all, we looked at the primary metabolites. So we looked at amino acids and sugars because they can both have positive effects. So we saw minimal effects of the infestation. However, we saw quite a strong effect on the protein levels. So Melodogena incognita had a positive effect on the protein levels in the taproot, so where the flies were feeding. We didn't analyze the fine roots. We only focused on where the insect is feeding. As you've seen here, there is 3 times we harvested at 24 hours, 48, and 120 hours after the root fly came on. And as you see here, always when there is a nematode, so the gray boxes and the green boxes are the ones with the nematodes, gray only the nematode, green with the root fly and the nematode. the protein levels are increased. So you see also here in the significance indicator that is a strong nematode effect on protein level, independent of whether you have a root fly or not. So this stays also when the root fly is feeding. Of course, it could be a very good reason, because we all know that insects are nitrogen limited. Protein is a very important building block, and especially in roots, nitrogen levels are even lower than they are in shoots. So this could be a very good mechanism over which these root flies, the nematodes promote performance of root flies. We also looked at defenses, and also here a very quick introduction. So Brassicaceae make glucosinolates. It's a typical flavor if you're mustard and you're Brassicaceae. So the glucosinolates are not the defenses themselves. Normally, there isn't a conversion by an enzyme called myrosinase, yielding, for example, isothiocyanates. Depending on the conditions, there can be also other end products. And these are the pungent flavors of Brassicaceae. And also, for example, wasabi. I once made the mistake thinking wasabi was avocado cream. Never made that mistake. First time I ate in a Japanese restaurant. Right, yeah, you all know the sensation. So it's a defense. So we also looked at glucosinolates as one of the defenses that are very important in this species, also in below ground, by the way. So they produce in both roots and shoots. So we looked at different types of glucosinolates. So we have indole glucosinolates and aliphatic, and why this is relevant, I'll come back to it later. So let's first look at the indole glucosinolates. Again, the controls are the gray boxes. We first look at the plants with and without the delia. The blue boxes are the ones with the root fly. Here you see some effects. So the root fly feeding increases glucosinolate levels, particularly the indole glucosinolates. But this is what we knew already. This is what we started off with maybe 20 years ago. So we're more interested what happens if we have a Melodogene there, and then we see that this increases even more. So you see here that sometimes also the induction is not so strong, but if there is a Melodogene there, it will go up. So there is a kind of a positive effect of a nematode on these kind of glucosinolates. So now we also looked at another pathway called the aliphatic glucosinolates, and these are different biosynthetic pathways. So one started with tryptophan, the indole started at tryptophan, and the other one started at methionine, which is another amino acid. They have their own biosynthetic pathway, so they can also be regulated independently. And there we saw that there was not so much induction by, so these are two aliphatic glucosinolates, gluconapin and glucobrassicanapin, which are very prominent. And there we saw that when there is a nematode, it decreases overall. There was a significant effect of interactive effect between the nematodes and the Delia radicum. And now you may think, listen, didn't she just tell us that Delia radicum is a specialist? So why would it care about having glucosinolates being, up or down or decreased or not? Well, this comes back, and that's also true because Rebecca's research showed that Delia radicum is able to detoxify these isothiocyanates that are formed. You see, different insects are very well able to tinker with this system, so they can diffuse this so-called mustard oil bomb, as it's sometimes called. So sometimes they do something with the precursor, something with the enzyme, or you just have to face the music and detoxify what comes. And Delica is one of them. It can detoxify these isothiocyanates, so it's very well adapted. So why do we look at glucosinolates then? Because they don't seem to care. Well, they do, and this is the research of Rebecca. If they are fat isothiocyanates, and here you see their survival rates, and this is a controlled diet. It's very difficult to get an artificial diet for these larvae, so we had a semi-artificial diet added with isothiocyanates. And you add isothiocyanates, like here is an isothiocyanate and 2-phenyl isothiocyanate, their survival decreases. So they can survive. Maybe other insects cannot survive, but it comes to a cost. And these products that they make, these amines, if you add them to the diet, it definitely helps. So the conversion products that they make help them to survive, but they cannot totally negate the effect of the isothiocyanate. So I think it comes all back to this famous saying, there's no such thing as a free lunch. So you they pay something to be able to occupy this niche and can detoxify, but it also comes to a cost. So it definitely is important that if you lower the glucosinolates, if the nematode lowers the glucosinolates, these organisms can benefit because to them it will be less cost to detoxify. So, and also because I saw 3 posters on Brassica napus and oilseed rape, we use also this knowledge to develop sustainable ways of managing this root pest together with breeders. So I just wanted to show you because yesterday I had very interesting conversations with several people also working on it. So yeah, so we now know that reducing these aliphatic ones like gluconapin can be part of this positive response of Delia radicum to nematode infections, but we still may be a bit puzzled about this indole glucosinolates. And the reason that indole glucosinolates, because we know also that indole glucosinolates, the conversion does not lead to these isothiocyanates, which are very toxic. These products that are formed, the indole carbenols and the acetonitriles, have been found to be less effective against insect feeding, but maybe they were very effective against pathogens. And then we started to think, hey, pathogens, because what we think is now what happens if you are having this below ground interactions with many of these interactions like insect herbivores and microbes and the nematodes, that also the insect herbivores never come alone. They will bring their gut microbiome, they open a wound, the wound may be infested with pathogenic pathogenic bacteria that are around in this rhizosphere. So we now are thinking, and Axel is working this out now, that maybe this response that we see in the indoles are not against the insect per se, but what comes after or what comes with it. So to take home is that that also below ground spatially separated herbivores can affect each other via induced responses. And that if you want to understand the value of each of these responses, you have to consider the whole complex, not only look at your herbivore, but also what comes with it, where is it, what else is in this environment. Otherwise, you don't understand this response happening. So yeah, I have now take a little bit of time. I'm looking at someone giving me minutes or so. No, okay, then I will just keep talking. No, not forever. There is an end to this. Don't worry. Now I want to take a few minutes to talk about the research of Jessel Bajaj. She is a PhD student. She will defend in about 10 days from now. And she was a nematologist, really interested also in this interaction between nematodes and aphids above ground. Yeah, we worked for the aphids, we worked with the cabbage aphids. I mean, here you see how beautiful it looks when your cabbage is nicely infested, apart from also the holes in there that probably appear as brassicae. Also here, it's I told you already, they bring effectors with them to establish themselves, but they are also highly adapted because they even can take up glucosinolates, they make their own morosinase, so they're walking mustard oil bombs. So they also use the defenses of their plants to defend themselves. It's also, of course, like any aphids, they can transmit viruses, which may be another problem. So when Jessel started, we already knew a little bit because I've been working on interaction between nematodes and aphids before. And we also already knew that different nematodes, so these root knot nematodes like Melodogina, would normally, there was associated with the induction of jasmonic acid, and they would have different effects than, for example, Pratylenchus penetrans, which was associated with the induction of salicylic acid. So their effects on these aphids were also contrasting. So Melodogina, also like it had on this cabbage root fly, it had more facilitating effect or also sometimes neutral effects, whereas Pratylenchus feeding usually had a negative effect because there was more defenses induced that would also affect Brevicurina brassicae. However, of course, we all start very simple. One nematode, one aphid. That's all good and well, but I also once did a little survey where I looked at Brassica nigra, which is the plant that Jessel worked with. And I saw that if I go to 3 natural populations close to where I lived and worked, yeah, that's usually how it goes, that if we look at the nematodes per plant, that we also, and which kind of genera are there, that we see that both are occurring, theirs can also co-occur. So it's never only one nematode. So there's different other nematodes that are belonging. There's always a community. So looking at one response of one herbivore to one aphid may be very useful for finding mechanisms, but it's not the reality that plants deal with. So this is why Jessel said, okay, nice that you did the one-on-one nematodes, and I'm going to work with two nematodes together and see what the effect is. And what are the underlying mechanisms? Again, we do like to also understand which responses are induced. And Jessel, who is also a very gifted designer of overviews, as you can see here, of setups, she also started with the nematode infestation, and then after 7 days, she started to inoculate the aphids. And then there was different moments in time where she harvested either the leaves, she looked at aphid survival, but she also, of course, collected the phloem. And this is not an easy task, you know, analyzing phloem. Phloem is it's not like a leaf is a massive, brassica makes massive leaves. So you have always enough biomass to do your analysis. But for phloem, it was really a challenge. So we also analyzed phloem because that's where the aphid feed. Yeah, so she looked at survival after 3 days after aphid infestation. So you have two timelines. This is the nematode infestation timeline, and this is the aphid. And then she also looked at population growth and again analyzed phloem. So we, at that time, it was at iDiv, we had quite a nice metabolomic setup where we could do LC-Q-TOF analysis. I will not talk a lot about the technology. And then you have workflows where you can identify what is in your sample. So let's first look again at the aphid performance. So it's from left to right, always the control plant, no nematodes, Melodogene, incognita, MI, I have this also here. Only MI, we look at surviving aphids. We have the only Pratylenchus, PP, and then we have the double one always in the middle. So you can always compare with the single ones on both sides and to the control here. Okay, so what she found first is that there is a significant effect of nematode infestation on the performance of the aphids. Pratylenchus, as before, was the worst for the aphid performance. Melodogene had no effect in this case. We found this sometimes a positive, sometimes a negative, and this is sometimes positive, sometimes no effect. And where there is two, they're kind of in the middle. So this is after 3 days aphids, and this is after 19 days, and we have a look at the population, and of course you have a lot of numbers to count. And then you see that there is a similar pattern, but it's not significant anymore. But for sure, the survival of the aphids was significantly affected. So now we would like to know what is in this phloem that may be different. Yeah, so we did metabolomics, and this slide is overwhelming. This is what I put up, like, we did metabolomics, look, we did heat maps, blah, 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 blah. But I'm not going to talk you to all of that because I don't have time, and it's also sometimes you need time to get accustomed to it. But I will focus on the first on the different in patterns and then on the up and down regulation. So I took out these figures here. So here we have a PCA plot where we basically look at the whole patterns of these metabolomes in the phloem. We take all the components and we ask the program, like, show us how these profiles are different. And the controls here are in black. This is this circle. This is the 95% interval. Then you have only the Melodogene is the red circle here. Then we have the Pratylenchus and the mixed ones. Yeah. In the leaves, we have a better separation, but I wanted to focus on the phloem because that's where the aphid interaction is. And you see that they are starting to differ. Yeah, so there is some differentiation in the chemical profiles of these phloem saps that we collected. Then we could also look at how many of these phloem components go up or down compared to the control in each treatment group. Yeah. So these are the down-regulated numbers of we call them features. It could be compounds, but we don't know what they are yet. So we call them usually features. So these are the down-regulated features in the 3 treatments, Melodogene alone, Pratylenchus alone, or the mix. And here the up-regulated. And what is then, it gives also information. Yeah, it's really very broad category information. But what you can see is that Melodogene mainly down-regulates these compounds, Whereas in Pratylenchus, you see a lot of compounds are upregulated, much more than downregulated. And also here you see that Melodogene is mainly downregulated, and Pratylenchus is not so much, and here it's upregulating, and Melodogene causes not so much upregulation, which is in line with this expectation that Melodogene, with its effectors, tries to tell the plant, no, don't kick me out, I'm harmless, downregulate your immune response. Yeah. By the way, just to tell you, that LCMS that focuses also on defense responses mainly, although you also find other compounds. Melodogene, the mixed treatment was somewhere in between. But we also found there is more overlap here in the response with the Pratylenchus than with the Melodogene. There seemed to be some either interactive effect. Like I said, metabolomics is hard to interpret. Usually, and I tell this over and over again, is a hypothesis generating technique where you say, okay, now I find something and I have to do the next step, which I also will show you later. Yeah, what we also found is that there is an effect on the sugar in the phloem. So here you always see the plants, again, the control, Melodogene alone, the mixed ones, and the Pratylenchus alone. Sucrose and glucose at 3 days after aphid, and this is after 19 days when the populations have grown. And what you see here is that aphid induced the sugar flow, they create a sink, but this is even larger if there is Pratylenchus or the mix of, although especially when Pratylenchus is present. And yeah, this is very interesting because if Jessel and I have a bet going on, Jessel thinks this causes a kind of an osmotic shock. I think, no, they don't care about it. They care more about the defenses. Because this is also what we found, is that if you look at other compounds that are affected significantly, and I want to just focus on 2 here, is that we also found there is quite a response in the synaptic acid. So Pratylenchus, where the aphids did worse, there's much more synaptic acid, and this is a phenolic compound that could be considered a defense. And we also found that it's very interesting that it's lower in vitamin C. Yeah. So first we thought, yeah, that's a vitamin, so maybe aphids need their vitamins too, and this is also have another effect on their performance, the lower performance on these plants. So this is actually this is the summary more or less of what Jessel also found, and also in other chapters of her thesis. So this is what we're now zooming into. And this is one of the chapters of her thesis that is not published yet, where she said, okay, if vitamin C, working from this hypothesis generating steps that we made with the metabolomics, then we're going to test this now in a mutant of Arabidopsis. And indeed, she found again that if you have a mutant, it's called VTC21, with lower vitamin C ascorbic acid, the aphid indeed performs worse. What exactly is the role of this vitamin C, if it's directly a vitamin for the aphid or something with the antioxidant response? This is all part of her chapter 3 in the thesis, and she's still working on it. But we're now trying to dive deeper into this aspect, just to say where this is going, at least for Jessel. So what is the take-home message here? So if you have double nematode infections, then you cannot infer from doing single experiments, single nematodes, what it would look like with the double one. So they ended somewhere in the middle, but there was also a lot of differences. It's very hard to interpret all these differences. So this is something that you then can test with single experiments again. And ascorbic acid or vitamin C seems to play a role in this response. The paper here is, there you have a QR code if you would like to follow. And just to make a little advertisement, this week we also just published another paper where it was about the 2 nematodes interacting via this root-induced responses as well. That just came out in Journal of Chemical Ecology, one of my most loved journals. So what did I want to send you home with before you take your train? So as above, so below. So we have systemic induced responses below ground, above ground, above ground, above ground, but also in the below ground realm where spatially separated herbivores or pathogens can affect each other. And if you really want to understand induced responses or how they evolved or how they work in nature or even agriculture systems, you have to go more complex. Yeah, it's scary and it's difficult. And as I said, if you then do metabolomics, you get even more complex. Of course, you have to then also do reductionist experiment again, but only by that. This is the way how we can make the puzzle. Yeah. So since I'm the last speaker on this conference, it's also my, what is this? I can also send you home with something maybe that you would not have expected, because we opened this conference with a remark of Wojciech about insects rule the world. Yes, insects maybe do rule the world, but they don't do that alone. They also have nematodes, and of course, there's many people that work on microbes. So if you don't understand something in your insect-plant interaction or your insects, anyway, maybe you have to also look at the nematodes, which are very colorful and diverse, I should say. They are fascinating organisms. And with that, I would like to show you all the people that contributed to my work in different groups, in particular the people here, April and Pius, that worked with Jessel on her papers, and Grit and Alexandra that were also involved. The groups that I worked in, and last but not least, you for your attention. I'm very happy to answer any questions you may have. Was I good in time? Yeah. Oh, 5 minutes.
Thank you so much, Nicole, for this awesome talk. There are any questions for the public?
Thank you, Nicole. Mine is a quick question. So did you look at the nematode dynamics on the plants and how, because if you have two different sort of genera on the plant, they sort of behave and reproduce differently. How do they do with competition between themselves? So I wonder if you looked at that or if you have any information about that.
Yeah, that's part of the paper that just was being published. Yeah, they do. Well, we added them at the same time, but there may be competition for feeding sites if they have to, compete for, because they all want to go for the fine roots. We checked if there was infestation and if this was consistent among the treatment. So this seemed to be the case. And there are some effects, but I don't think they were very strong. And this is also a fact of life, yeah. So this is part of the complexity of the system. But yeah, there's definitely effects, because they can also affect each other via induced responses, or being first or being second is also an important aspect of it. And there is behavioral elements where they attracted or not attracted. Yeah, you're completely right. But overall, we saw that was rather in the treatment groups, we took them as they are. We just checked if they were there, and they were all there. So we used molecular markers to check if the roots were infested properly. And of course, later you can see the root knot. So Jess always had an extra plant on the side to monitor what was happening below ground, because you simply cannot see it. Yeah, but you're completely right. One should take this into account as well. Yeah. Already. Okay.
Hi, thank you for your talk. Do you have any idea of how the symbionts of the aphid could impact all this interaction? This is another level in complexity.
But no idea whatsoever. So anyone could say. We didn't check. We had, of course, in-house cultures, that were derived from singular females. So they're quite uniform. Indeed, you can add more complexity. How does it affect? We know that it sometimes also affects the higher trophic levels. Yeah, it's an endless puzzle that we're making, but it's fascinating. Yes, but good question. I don't know.
Online question from Victor Sorara Carrasco. Have you looked into the effect of plant responses induced by insect infestation on nematodes?
Very good question. We know that it can have effects, because we know that it also the responses also can go down, especially when you think about root exudates that nematode use to find their plants. We didn't do the whole because, as I said, we start with the nematodes because we think that's also the normal sequence of how their infestation is. Then we should have looked at, I'm trying to think, no, that was also, we also looked at the timing of the life cycle, but it wasn't tomato and the effect on above ground, but not the other way around yet. But I'm sure that also has an effect, but but I don't think above ground nematodes come before the, above ground herbivores come before the nematodes in this case. But of course, if you're thinking about multiple cycles, because these nematodes I think they take 6 weeks under warm conditions to fulfill one cycle. So they're in a normal season, they might be an effect on the second generation. Yeah, more to do. It's good to be a biologist. We have always work. There's always more questions.
Yes, there's always more complexity, and I'm going to make a vote for the plants that have to deal with all of this. They may not rule the world, but they're doing a good job. I've really enjoyed your talk. Thank you so much. The first experiment case study you presented, if I remember correctly, you said the infestation with nematodes was perhaps at the lower end of the population densities you might see in the field, and that had a positive effect on the cabbage root fly and also on root protein content. And it made me wonder whether there might be dose-dependent effects, that if you had a higher abundance of nematodes, would it enhance the effect, or would it mitigate the effect? Is that anything you could comment on?
We took the densities that I found. We took the numbers of nematodes to inoculate such that we would add up with what we call the natural of the wild population densities. If you look at people that look at plant pathogenic nematodes in agricultural settings, of course there is also a build-up of nematode population densities on fields that are being used over and over again for same crops, or at least crops that are similar enough for the nematodes to multiply on. And there, the densities that people use to infest one plant are, for example, 2,000. And we started with 400 because that worked for us, achieving this infestation rate that I found in my wild populations of Brassica nigra. I know that, for example, if there is, for aphids, I know this, but I assume it might be also true for nematodes, that if there is an overwhelming number of infestations, at a certain point, the plant collapses. And then the effects, of course, may be different. Yeah, we have not studied that, but I could imagine, and if you read the literature, how devastating nematodes can be. Sometimes you can see them in fields. You can see where there is a patch of nematodes just destroying the crop. Yeah, so then the aphid, of course, there's nothing to feed on. So whatever nematode it may be, it is another effect. We just looked at the responses, where there is still a plant that can respond. Yeah.
Sorry, all I was going to say, it might also be interesting to look at the lower end. Like at what threshold do you actually start to see an effect?
Yeah, that would be really interesting. And actually, the first experiments we did with the single nematodes infestations, I was a student from Ghent. He's a nematologist, and he also looked at the behavior of the aphids are attracted or not attracted to nematodes, that plants that were good for them, so the melodogene ones versus the one with Pratylenchus. And at a certain point said, oh, we should, at his defense, it was a bit funny, I had to laugh. So it's like, oh, maybe we should we should tell farmers not to worry about Pratylenchus anymore, because they are, they will prevent aphids from growing. I was like, yeah, but at a certain point, you also have a problem with your nematodes, yeah. But yeah, this is the complexity of things, yeah. And this is also what we have to deal with in agricultural settings. If we want to reduce the pesticide use, that we may have to become, and especially farmers have to become managers of complexity. And this morning we also had this, like, where is the threshold for spraying against aphids? These things, maybe we are very strict at the moment, but we should maybe think about this. But it's really hard. It's a tough message to sell. Yeah, very tough message. Yeah.
Time for one more question. Yep.
There's one.
That was a great talk. Thank you. Did you notice any behavioral changes in the feeding of the herbivores in response to nematode infestation?
The behavior of the aphid feeding, or the root fly we cannot see. It's below ground, so we could make a see-through pot, of course. What we saw with the aphids, that was in the earlier experiments, that they start to if you give them a choice, you have 2 leaves. We didn't do this in these experiments, but you have 2 leaves and you put them in a petri dish for them to choose with the plant still attached. Or actually, I think it was a little bit of a paper bridge, and you put aphid in the middle, it will start to choose for the plant that is where the immune response is a bit suppressed. So apparently they probe And then they assess, oh, it's too difficult here, try the other one and settle there. So that we saw that there is effect on the behavior. If you want to know about the feeding behavior, I'll have to do that. You need an electropanotogram or panetometer. Then you can maybe also see that they start to in the plant, you know, that we couldn't do because we don't have that. But for the choice behavior, we saw some effects in the single ones, yeah.
Okay. Thank you so much,
Thank
Nicole,
you.
for
Yeah.
your talk.
Could the student reps come up here, please?
Not just me.
Can we just share the main desktop? How do we do that? How do we share the desktop? Sorry.
It's not possible to share with Dexter, so you might have to open the slide and then share that.
They're not slides, they're just the next pictures and everything like that.
Are they on the computer? Okay. Which one?
But we want to open one at a time.
So you might have to open and share each of them.
Okay. Don't want to cover the screen. Don't look.
Yes.
Can we share this and it'll just show the documents they open on the.
It's just showing the whole screen up there. I'll be doing it on.
This does not allow.
Okay.
Oh.
Yeah. It doesn't allow. It doesn't allow to share the screen.
Okay.
Okay, let's see. Okay, you want to share? So probably when I don't know.
It shows us the screen that we're sharing.
Yeah, so we can see.
So you try and see.
Okay. Do this one. You'll hear me? Is that good? Hello, everyone. We are your student reps. My name is Ben. This is Rosie, and our third student rep, Idris, couldn't join us today, unfortunately. But we just want to plug very quickly that we have a vacancy for another student rep. So students, look out. Well, it's online now, so please apply. And
Because Idris is missing.
it's not because Idris is missing. We're not looking to replace Idris. But yeah, we were tasked with judging the Ento fashion, and you didn't make it easy. My goodness, you looked great. Yeah, so we were taking photos of people around yesterday. You may have caught us, with permission, obviously. And yeah, we managed to choose some people as our winners. We'll announce the names, but you can stay seated. Just please come and find us after at the res desk to come and collect your prizes. I think that's everything from me. So over to Rosie.
It was quite controversial. We actually disagreed on some, but we managed to come to a conclusion. But we wanted to do some honourable mentions first. So we really liked Richard Harrington's whole butterfly ensemble yesterday. It was brilliant. And we really want to give a shout out to Claire Boys and her brilliant brooch that she made herself. Now for winners, which...
The prizes.
Oh, prizes first. So third prize is a really lovely scarf from the Roving House. It's a myriapod. I'm not going to say if it's a centipede or a millipede because I don't want to get yelled at. It's not my specialism. But for third prize is Linda Birkin. There you go.
Yeah. Okay. Take a break.
Okay, and our second prize is for Cynthia Oy. We really loved all the accessories. Oh, sorry, I didn't say what the prize was. It is a bug box from the roving house, which includes like all of the pins and stickers and some bobs.
Okay.
And then our first prize, which is £500 worth of clothing vouchers from, is it Paramo? Paramo. Was Holly Bassett with her great ensemble. Oh, it was a video, but we don't know if it's going to play.
Yeah. Oh my God.
Go, go, go.
Once more.
Everyone online gets it slightly slower. Go. There
we go.
Yeah, it's so cute. Ben and I are all right entomologists. We're not good people.
Yeah. Thank you. That's all.
Yeah,
from us.
thank you.
Well done.
Thank you very much. And it's my pleasure to close out the meeting. And Ben, don't get comfortable, because the first thing I'm going to say is a thank you to Ben for being a student rep. And so we do want you to come down here. And we have a little something to say thank you for being such a fabulous student rep. So if there are any students out there that would like to take on the role of student rep, then you can ask Ben or Rosie what the role entails, and please do sign up. So thank you, guys, and well done for the Ento fashion, and congratulations to our winners. We're coming towards the end of the meeting. I have the pleasure to announce the poster and talk prizes, and I will also say a few thank yous. So I'm going to present those prizes first, and we do want you to come down, and we do want to take a photo as we present you with those prizes. So I'm going to start with the best talk prize, and this prize, do you want the drum roll thing? Thank you. The best talk prize goes to Holly Bassett. And the best poster prize. Thank you. Best poster prize goes to poster P31, Carmanye Chaduri. I'm sorry if I pronounced the name wrong. There. And last but not least, the best online poster prize. And I'm assuming, oh yeah, please. I'm assuming this person isn't in the room, but perhaps they are. But it's online poster number 3, Silas Biggin. Well done to all of our prize winners, but more than that, well done to everybody who presented over the course of the meeting. I imagine that was actually difficult getting to a final list of prize winners, because there was just some outstanding contributions in terms of talks and posters. So thank you all very, very much. There's a feedback form. Isaac, is there a slide for the feedback form? So while we're finding the link to that, brilliant, thank you. So please do fill in the feedback form. It really helps in terms of making sure that our future Ento meetings are the best that they can possibly be. So please do fill in the feedback. And It's a good opportunity while you're doing that to thank our student helpers who are here in the front row in the blue t-shirts, who have been absolutely brilliant at supporting over the last 3 days. I don't think I've ever been at a conference where the AV technical support and support from the conference helpers has been quite so good. So thank you all sincerely very much. As we mentioned future Entos, am I allowed to say where it is next year, you know? Yeah, okay. So we hope that you will all join us at Ento 2026, and we can announce that that will be held in Cardiff. So we hopefully will see you all in Wales this time next year. Before then, those of you that were in Gwen's talk a moment ago will have heard her mention the European Congress of Entomology. So at the end of June, beginning of July next year is the European Congress of Entomology, and the RES is supporting that. And it's in Tours, in the Loire Valley, in France. And on their website, they say there will be outstanding science, but also a lot of really nice cheese and wine. So if you're not going for the insects, then come for that. I just want to thank our fabulous conference conveners. Fiona Hyatt, Ali Carney, Gail Jackson, and Maro Pasmino, thank you so much for putting together such an amazing program. I think we can all agree this has been an incredible few days of insect science. And also thank you to our plenary speakers. So thank you to Wojtek Nowotny, Christiana Verauch, May Berenbaum, and Nicole van Dam. And again, I don't think I've been at a conference before where all of the plenaries have been so fascinating and so interesting and so incredible. I feel like I don't know what I've been doing with my scientific life. You guys are just amazing. So thank you so much to those for those plenaries. But again, all the presenters and also all the workshop conveners. The workshops I attended were great fun, as well as being informative. So thank you to those who convened workshops. Thank you to the Glasgow Convention Bureau for their support. Thank you to the University of Strathclyde. I think this is a fantastic venue. So it's been great to be here. Thank you to the exhibitors. Hopefully you all managed to get to see all of the exhibitor stands over the last couple of days. And I don't want to forget to thank the online attendees. It's difficult when you're in the room to remember the people not in the room. So thank you all so much for taking part. It's not always the same as being in the room, but hopefully you managed to enjoy the presentations and the parts of the conference that you were able to attend. You missed the dancing last night, so a particular thank you to our local conveners for a very enjoyable conference dinner, and Kaylee, and I'm not sure my legs are going to recover, but it was great fun. Thank you for that. Sponsors, I'm nearly there at the end of the list of thank yous. Sponsors, thank you to Darwin Biological for the support for the lanyards, to Bartlett Tree Experts and Harper Adams for supporting the plenaries. And my final thank you, I think, as long as I haven't forgotten anyone, my final thank you is to the fantastic RES team, led brilliantly in terms of the conference organization by Fran, and in terms of everything else by Simon at the back there. Really, you guys have been fantastic. Thank you so much for all of the work over the last few days, and not forgetting poor Bianca, who has been organizing all the online attendance. And so Bianca has been sat online on Zoom for the last 3 days, managing people in and out of rooms. And I think that's possibly the most boring, but a very worthwhile job. So thank you to Bianca for that. So I think that's all of my thank yous. Have I missed one? I forgot to thank Forest Research. And we need to announce, is there a treasure hunt winner? Who wants to talk about that? Okay, sorry, Anna.
Thank you. Thanks, everybody. And yeah, well done to everybody who entered. Thank you for your enthusiasm completing the quiz that we had round. The answer to the secret word was spruce, so well done to everybody who got that right. And we've got 2 winners of the insect rucksacks. And if they pop by the FR stall just after this, then we can give them to them. And the 2 winners were Anna Evans-Wolf and Zuzana Deblova.
Thank you, Anna. And so I think that's everything I was supposed to cover. So before we officially close the meeting, I'd like to ask the local conveners to come and have the last word. Thank you.
Hello, everybody, and thank you so much for coming all the way to Glasgow to join up with each other. Glasgow's theme at the moment, I think, is the people make Glasgow, and I think between the buzz in the room every day and the moves on the dance floor last night, I think you guys have really done it. And yeah, and yeah, I'm sorry, everybody online, you have missed out a little bit with the dancing. Our job has been really made so much more easy by the Royal ENTSOC community and the team behind all this. Really, they know what they're doing. They've done absolutely amazing. And we just wanted to say a special thank you to Fran, who has guided us through this. We had no idea what we were doing. So
and we wanted, I know this is your first Ento, and you've been incredible. I mean, nobody in this room would guess that this is the first time you've organised this conference. And on the day yesterday, when we were all wearing our entomological attire, you were sort of partly apologising for the fact you didn't have something hugely entomological. So we've decided to rectify that. And next year, you have another one.
I think that's really all from us. There's a Scottish saying that says, haste ye back. So hopefully if you've enjoyed yourself and you haven't had enough time to see Glasgow and Scotland, you might come back another day. But thank you.